All right. Hello, everybody, and welcome. So pardon my bandages. I am moonlighting as a mummy. So, as the great philosophers, the Wu-Tang Clan once said, cash rules everything around me. Cream, get the money, dollar, dollar bill, y'all. All right. So we're adding a little bit of flavor to the seminar today. So I first met Dr. Burton at the Keystone Symposium on Pain and Somatosensation in early February of 2020. And this was during which time I was applying for a faculty position. So in the US, we recall this time as BC or before COVID, right? So this was about a month before everything shut down. So I was fortunate to be able to meet him during that time. So the first thing he did was he gave me a giant bear hug. And so the New Yorker in me immediately sort of short-circuited, was like, oh my God, what is this? But yeah, it's always a pleasant surprise to meet someone who is so talented, but who's also so approachable. So now for the accolades. Dr. Burton received his BS and PhD in animal sciences with a focus on immunophysiology and behavior at the University of Illinois at Urbana-Champaign, Chi-Town. He then completed his postdoctoral work in the Department of Hypothalamic Research at UT Southwestern Medical Center, and then in neuroscience at UT Dallas, where he worked with Ted Price and Greg Dussor, and he has since distinguished himself in his own right. He received a highly competitive K-22 NIH Transition to Faculty Award. Currently, Dr. Burton is an assistant professor in the Department of Neuroscience at UT Dallas School of Brain and Behavioral Sciences. He is a founding member of the Center for Advanced Pain Studies and the director of the Neuroimmunology and Behavior Lab. Dr. Burton is an author on nearly 70 publications. As an assistant professor, you're making us all look bad, including several promoting diversity, equity, and inclusion in STEM. He is a member of the CSR Advisory Council Working Group at the NIH to institute changes for future fellowships, future F fellowships to increase diversity. He's a recipient of several prestigious awards, including a certificate of congressional recognition, as well as the Rita Allen Pain Scientist, Mitchell Max, Keystone Symposia Fellow, and Future Leaders Award from the American Pain Society and the Endocrine Society, among other things. His work has shaped our understanding of how sex differences and effects of aging on immune system and metabolic processes governs how acute pain becomes chronic. And you may or may not know, but chronic pain affects 33% of all people. That's more than cancer, diabetes, and heart disease combined. He is also a formidable advocate for URMs in science, a subject which is also near and dear to me. And he's a fantastic collaborator and a dear friend. It is my distinct pleasure to introduce you to Dr. Michael Burton. I look forward to see what he does in the future. I'm very excited about it. The title of his seminar is Neuroimmune Crosstalk in Acute TLR4 Induced Inflammatory Pain. Dr. Burton.
Awesome. Well, that was quite an introduction. Wow, that's super lovely to hear that. And I just really appreciate Ann and Matt for the invitation and the opportunity to come and speak with everyone about some of the research that my lab's been doing. Yeah, I always forget some of that stuff happened in your life because things are just flying by. Time is just flying by. But okay, so let's kind of jump into it. Double check, make sure everybody can see this. Cool. All right, so as I was saying, my name is Michael Burton. I'm an assistant professor, and we run the Neuroimmunology and Behavior Group at the University of Texas at Dallas. I always like to start off talks by talking a little bit about myself, as you need to hear more, right? At least where I came from, and then put it into some context about why we're doing it and why is it important. So first and foremost, she said Chi-Town because I am from Chicago, born and bred through and through. I went to the University of Illinois where I received training in psychoneuroimmunology. That is how the immune system and the nervous system interact to essentially mediate pain, well, mediate behavior. And so we'll say adaptive behaviors that are important for sickness. And then went to UT Southwestern and received training in endocrinology and metabolism. And then UT Texas at Dallas, where I received training in neurobiology. So there's kind of 3 different realms of training that I've gotten. And so now my expertise is in places that are really integrating at a really good time right now. And so that's one of the reasons why the lab's able to be as productive as it is, because we're able to talk about things that people hadn't talked about before. So my lab's in this building. We started in 2017. It's in the basement. So you see this really beautiful building with all these beautiful windows. When you come into my lab, you expect to see that, and you're like, all you see is walls, because we're in the basement next to a bunch of mice. But that's where we want to be, right? We want to be right next to where the animals are. It's a beautiful building. It opened up in 2016. We slid in right, the next semester after it opened up, essentially. And so this is kind of where us and the mice live. And we are part of this greater Center for Advanced Pain Studies, where myself, Ted Price, Greg Dusor, founding co-directors or co-founding members. And this is, love to show the picture. Obviously, you can see all the trainees that we have, very, very diverse. It's a really beautiful place to work. And at this point, we've gone from 3 faculty members to 17 now. And it's, obviously would need way more space to take an up-to-date picture. And, we always talk about some of the funding that the center has, which is from the Rita Allen Foundation and NIH as well. Okay, so then going back to me and my lab, I always want to kind of recenter. So we have these expertise: psychoneuroimmunology, endocrinology, metabolism, pain neurobiology, and really taking integrative approaches and understanding how these discrete systems work with one another, communicate with one another, and then really developing biochemical bases of this interaction and communication, because physiology is way more complicated than just one box. The immune system does this, and that's all it does. The nervous system does this, and that's all it does. Well, they need to talk to one another to make sure that everything is where it needs to be. And so that's what we are at what we do. And so current projects in the labs are 4-fold right now. Used to be more, but we had to tone it down. COVID was really obviously not good for a lot of different reasons, but it did make us more focused. And so I'll tell you 4 that we're doing, and we'll just talk about one. So the first project is looking at aging, using an animal model of aging and understanding how age and sex influence the immune system and different behaviors that kind of come from that. And there's a lot of young people in the audience now. Maybe you don't think about that now, but talk to me in 15 years when things start to creak. Then this starts to, and people in the back may know. So this is obviously something that everybody deals with, and we feel there's a really strong and approachable model, and hopefully has some longevity in the lab. This neuroimmune communication project is what we'll talk about today: how peripheral neurons communicate with the immune system to mediate pain states, but other comorbidities like depression and anxiety, which we won't talk so much about today. We have actually various models now, cell-specific models and TLF4 signaling, chronic muscle pain models and fibromyalgia models, and clinical outcomes. And so we have both animal and patient projects in that. And then we're interested in the role of cannabinoids in mediating affective behaviors in analgesia, and really what cell types are most important to differentiate an effective behavior over analgesia, and potentially using cannabinoids as an opioid alternative to chronic pain. And this is a novel animal model that we use for this. And we'd love to talk about that if any people are interested later. And the last project is the role of neuroimmune biomarkers to indicate pain, post-operative pain, delirium, and convalescence after a complex neurosurgery. And this is in patients. Have a really awesome collaborator that's a neurosurgeon that's like super interested in trying to understand how some patients walk out the clinic after this complex surgery in 7 to 10 days, and other people take convalescent homes and need 2 months to walk out. So we have some really cool stuff we just started cooking up last summer when we opened back up. But again, today we'll talk about specifically the TLR4 communication project. Well, and I already mentioned it earlier, why do we care about pain? I have this slide that I always have to talk about no matter where I go, who I talk to, so people can get a feeling and kind of bring them into our projects. So billion effects, our pain affects 1 billion people worldwide, from chronic to acute pain. Of those numbers, a disproportionate amount of those people are women. And this affects quality of life, from daily social interactions, work, home life, costing roughly $635 billion a year, lost wages, medical costs, et cetera. So clearly there is this financial burden that is huge. When I would give this talk even a year ago, talking about the previous year, the idea of the opioid epidemic has been around for a little while. And 2 years ago, these numbers were 68,000, 74,000. And then this was published in the New York Times in just February last year in 2021. Over 101,000 people died from last year from opioid overdose because of the opioid epidemic. And that was kind of something that I didn't expect, right? You would think people were gonna be inside, not be so interactive and have different opioid experiences, but that flipped on us in a way that we didn't even imagine. And really it's about 40% higher than it was the year before. And so obviously this brings along, amongst the bigger pandemic, the epidemic of trying to understand different and efficacious non-opioid based therapeutics for the treatment of chronic pain. There's huge initiatives by the NIH for this. HILL initiative is one that comes to mind that has committed millions and millions of dollars to try to figure this out. And so for our lab, and in our lab, we want to understand how the mechanisms of pain signaling differ between the sexes. And that's because for a very long time, only males are used in pain research, and it was kind of out of focus, meaning we were finding out things that were in males, but maybe are different in females. And I'll show you today, definitely are. And when you do a preclinical trial or preclinical experiment and focus on one sex and try to understand what's happening in both sexes in the clinic, and it fails, like 80% of things do, people lose a lot of faith and hope in preclinical research, and it's really, really frustrating for us who do a lot of this research. And so one of the things that my lab and a lot of people do is try to focus on both sexes, but also try to bring in human or clinical aspects to the preclinical realm, just so that the relevancy stays there. So this kind of thought brings on a transition of a type that's super important. And so there's a classical thought that the immune system, we'll just say over here, and various immune cells, mast cells, macrophages, et cetera, interacts with the peripheral nervous system, particularly a peripheral pain cell or nociceptor, to sensitize that particular pain or nociceptor to cause more pain. So there'll be some type of stimulus from the periphery where these cells, these immune cells are affected, release these factors. The neurons have the receptor for these factors, and that leads to the upregulation of intrinsic ion channels that make animals and people more sensitive to heat or touch or other negative, well, we'll say changes to sensitization, right? So in the context of sometimes a stimulus may be innocuous and it becomes painful. Sometimes the stimulus may already be painful and become more painful, or hypersensitivity. Either way, you just have more pain because of this indirect activity between the immune system and the nervous system. All right, so fine and dandy. I was a postdoc at UT Southwestern learning about how peripheral sensory neurons affect metabolism. Turns out not a ton, they do, but they really affect gut motility more than anything else, primarily. And there's probably some other things that they do, but right now that's what we're figuring out. And I was a postdoc, and my projects really weren't moving forward. And in 2013, this paper came out from this group at Harvard. and discussed how bacteria directly activate sensory neurons to modulate pain and inflammation. And before, remember, the thought was bacteria interact with the immune cells do this, and then et cetera. But no, not at all. They tagged some sensory neurons with this red fluorescent protein that has a particular channel 1.8. And then they found that if you injected a fluorescent bacteria, that they interact directly with the sensory neurons. And they did it in a couple of different fashions. They did red and green for the neuron and the bacteria respectively, and then they flipped it, and you could see that there is a direct interaction between the two. And through the experiment at the end of it all, they did a ton of different things. This is a Nature paper, right? So like 54 figures with 54 subpanels, right? How that works, when we were talking about this earlier, Matt, right? How many millions of dollars, right? But at the end of the day, you take out and think about tissue swelling, immune cell number, pain, and bacterial load. So the amount of bacteria here in black actually correlated directly with the amount of pain that the animals were feeling at the time. And if you were to ask a clinician even before this or exactly at this time, they would probably shift that over to the pain is directly related to tissue swelling and immune cell number, actually. And so when you kind of break it down into hours like this, and this is, 2-3 days, not necessarily over the course of a week or something like that, and have control over how much the bacterial load is, you can kind of log it and really see and understand that it's not necessarily this indirect activity, it's a direct action. So again, this brought upon, well, sensory neurons are doing a lot more than we think and interacting with our environment in ways that are a little bit atypical. And so this paper found out that how these immune cells interact with bacteria is the same way sensory neurons interact with them, and they have a subset of similar receptors, and toll-like receptor 4 was one of the receptors. In this paper, it wasn't the most important, which was great because we had a model that was toll-like receptor 4 specific, and we knew that we can work with it. And so that's what we did. Okay, so what's the purpose of the experiments and exactly what we're doing and why? Well, so first and foremost is to understand, well, I said acute phases, but really is to understand all phases if toll-like receptor 4 is responsible in all phases of different types of pain. So if it's the acute phase, the chronic phase, and importantly, we have this phase called acute to chronic transition, and the output that we use is called an assay called Hyperalgesic priming. We'll talk quite a bit about that in a little bit here. But so the machinery or pain plasticity of machinery that's involved in this transition from acute to chronic pain, we believe or hypothesize that is one of the most important concepts to try to mitigate adaptive to maladaptive pain, right? So we want to know if you have a pain state, it's adaptive, I stub my toe, it doesn't feel good. I need to know that. I need to know that there's some tissue damage. If I hit it again, it's going to be more tissue damage. So I need to avoid that. And that's adaptive. I need to do that for a little bit of time, and it needs to go away because I can't continue to feel pain once there's resolution of the wound. But in some cases that doesn't happen. And then you get this chronic pain state that is now negative or maladaptive, and you want to seek attention for it. People seek attention, start taking different types of meds. Opioids actually have a variety of interactions with that type of behavior. You have some people that take opioids over a period of time and it does nothing, right? All of a sudden, I was actually talking a little bit just ago about different levels of opioids and how some people it will be very helpful, some people it doesn't help at all. And that leads to some people seeking more and more potent opioids and leading to overdose and overdose deaths. So trying to understand this machinery is super important. And lastly, what are the cell types that may mediate the phases of pain? So acute, the transition to chronic, and then chronic pain. Because not all things are the same, we imagine that immune cells are still important for sensitization for long term. And sensory neurons particularly are going to be important for both acute and transition, acute, transition, and chronic as well. Okay, so there's a couple papers that were kind of important after that, the first one I was just talking about from the Harvard group, and this one in Nature Neuroscience was talking about how different immune cells regulate mechanical pain or pain hypersensitivity in males and females. And so they found that particular paper found in a neuropathic injury, microglia or immune cells in the spinal cord mediate pain in males, in T cells in females, and it wasn't any other explanation. And something else we were talking about earlier, there's no other alternative. And the same group found that if you look directly at the microglia and look at the toll-like receptor 4 pathway, that was what really was distinguishing the difference between males and females. And if you did a specific assay in the spinal cord, you can see that males responded and females didn't. So, all right. What is toll-like receptor 4? And I gave a talk yesterday and or the day before yesterday, and no, it was yesterday, and it was to a group of people that were like, they knew all about toll-like receptor 4. Someone even said it in the chat, we know enough, you can skip this slide. So hopefully, yeah, that was fine. So, but anyways, and I'll make it quick. Toll-like receptor 4 or TLR4 is a molecule that was initially discovered to be only on immune cells, but now we find that it's on a percentage of peripheral sensory neurons, not central neurons, stromal cells like fibroblasts, epithelial cells, on a multitude of cells. And what the responsibility of toll-like receptor 4 is to recognize a pattern of a molecular pattern. And so in the I guess in the wild, you have something called a pathogen-associated molecular pattern, which is on bacteria. And the bacteria cell wall is really what consists of something called lipopolysaccharide, which essentially is a string of fats and sugars. And they bind in a motif to TLR4. Interestingly enough, our body produces something called danger-associated molecular patterns. Our body meaning pretty much all cells, which have a similar fat sugar backbone. And there's also heat shock proteins that have a very similar backbone as well. We also find it in short chain fatty acids as well. That's exactly what a short chain fatty acid is. And so they also interact with toll-like receptor 4 as well. And so the idea is if you have TLR4 and a nociceptor, a microglia, or a macrophage, and a fibroblast, which one of those cells are mediating or helping to mediate these phases of pain? So the sources of pain that we study in my lab are some acute inflammatory models, like using a toll-like receptor 4 agonist, some other inflammatory agonists. We do some long-term inflammatory models, chemotherapy models, diabetic model, and 2 different types of diabetes models, and then surgically induced neuropathy models. We've tried most of those. And just to get an idea of the translatability and understand how various factors like emotional, biophysical, psychological, so higher brain regions, interact and comorbidities interact with nociceptive input to mediate the emotional and pain response in general. And trying to get as close to a translational model as possible, right? So turns out that even some of these, like for instance, this fibromyalgia model is nowhere near reality, but it's super interesting still. And so we really, really like it. Versus using like some of the diabetic model stuff is maybe a little bit further off than what we wanted it to be in the translation. Anyways, okay, so for that, we also combined all those different kinds of models with genetic models. And so today we'll talk, hopefully we have enough time to talk about 2 different models. And turn on my timer. Where there's a requirement model and a sufficiency model for toll-like receptor 4 on either nociceptors or macrophages. The requirement model is essentially a Cree-lock system. You add a LOXP site to something, knock it out. The sufficiency model is a little bit different. It's kind of like if you've used a tomato, use like a GFP Cree line where there's a transcriptional blocker in the middle of the gene, so whole body knockout or null, and then you can cell specifically reactivate that particular gene in a particular cell type. And so we have both to talk about today, not a ton of data, but just a little bit to kind of convince folks that it does work and what we're finding is real, and hopefully the world will know and we can finally get this paper accepted. This has been a love child of mine since we opened the lab in 2018. And so, and then we want to understand acute or onset, translation or transition, and then chronic and chronicity in all these models. So in order to do that, we use this danger-associated molecular pattern. We started to use this danger-associated molecular pattern, HMGB1. And where I gave my talk before, I decided to keep it in here because there's aficionados that talk about different states of HMGB1. It's redox dependent. So you have an all-reduced state, oxidized state, and a disulfide state. And the different states respond to different receptors. And so we use particularly the disulfide redox dependent state, HMGB1, injected into our animals to see what kind of pain does it elicit, and is there any differences in males and females in general. And the reason why we wanted to do that is because we see that HMGB1, a danger-associated molecular pattern, is up after injury. It's up in quite a bit of the models here in chemotherapy. It's up in fibromyalgia. It's up in neuropathic pain models as well. So let's use the source that's TLR4 dependent and inject it. All right, so jumping into a little bit of data. So this is what we call mechanical hypersensitivity data. On the y-axis is something called Paul withdrawal threshold, which is essentially the higher you go, the less pain or less sensitive the animals are in, because it takes a little bit more gram threshold for them to respond. The lower you go, the less gram threshold response and more pain the animals are in, and people too. And so we come in and do a baseline for all groups, and then we do an injection right after the baseline, and then record for about 24 hours, 40 hours, and then I'll tell you about this in just a second. 3 different doses: a vehicle dose, a 0.3 microgram dose, and a 1 microgram dose. This is all for literature. And we're just kind of, we're doing some repeating experiments just to be sure that it's real. And we did it in males, we did it in females, and we saw that independent of the dose or dose responsively, that HMGB1 caused acute pain states, and the animals went back to normal or baseline after a day, 2 days. We initially, we put it all together and it was just all clustering together and there's no difference between the two. And so we've been showing it now separate, but we have put them together. There's no difference. Here is one of my favorite experiments that we do, which is this hyperalgesic priming experiment. And I probably should separate it out because it's really cool to talk about by itself. So if I think about the machinery involved in the transition to chronic pain, it is essentially taking a neuron and having a neuron become more sensitive across time. So in a typical PAW or typical situation, you inject some subthreshold dose of a mediator. This group, John Levine's group, developed this model some years ago, and you use something called a subthreshold dose, or really, really low dose of whatever you want to use. Actually, it turns out that a number of different things work, but they use prostaglandin E2, so we decided to use prostaglandin E2. When you inject a low dose of it, I think is 100 nanograms in the paw, you get this, initial response that lasts maybe half an hour because you injected something into the paw, but it doesn't do much of anything. However, if you had some prior priming response like carrageenan or HMGB1, where the animals were injected, allowed to go back to normal, but however, this transitional machinery is still active, causing sensitivity, this low threshold now causes this exaggerated and protracted response, meaning you see this hyperalgesic response. So if we go back, the animals after HMGB1, they go back up to normal after about 2 days. This is 16 days or 2 weeks essentially after the first injection. And then we inject them all groups, so vehicle in both doses with prostaglandin E2. And you find is if they didn't have a response initially, they don't respond to the prostaglandin E2. But if they did, there is a dose response that was dependent on the HMGB1 response and not on the PGE2 response, right? And in males and females, you see that both sexes respond in a dose-dependent fashion. So there is hyperalgesic priming or transition to chronic pain and HMGB1 injections. And so that was kind of the new thing that the lab contributed to the HMGB1 realm. Other people have gone through and injected this kind, that kind, and saw it did nothing unless you injected absurd amounts into the animals, et cetera. But this dose is pretty low and really indicative of what we usually find after like a major injury. We also wanted to do a little bit of inflammation or paw edema. So we looked at males and females to see if there's any differences in like inflammatory processes. So long story short, we looked 4 hours, 24 hours after these injections and didn't see many differences from change from baseline, meaning that there's inflammation in males and females, and it doesn't seem to really be different. Not truly different that we would expect. So than what we would expect. So kind of moved on from the inflammatory component and wanted to say, all right, so let's take TLR4 away from different cell types to try to understand who's doing what in the process. So to target TLR4, take 2 LOXP sites around, I think there's only 4 exons in TLR4, and you LOX or FLOX 2 and 3, and then you get mediated recombination, and you get that delta for that particular group. The genetic tools that we use, or the models that we use, were 2. 1, NaV1.8 Cre, that's important for sensory neurons, and lysem Cre, that's important for macrophages and granulocytes. And down here is just some validation data. When you have the toll-like receptor 4 by itself without the Cre, it's in about 30% of the neurons, which is super important in a longer conversation we can have later. And then when you cross it with the NaV Cre, you get a significant reduction. And the same thing happens when you have lysozyme Cre versus not. You get a significant reduction. This is gene expression from peritoneal macrophages. So both of the models work in their own right. All right, so let's go back to our handy dandy mechanical hypersensitivity data. And so these are the animals that have toll-like receptor removed from their macrophages. Black and gray are essentially the vehicle animals that have either FLOX or the lysozyme Cre removed, TLR4, and black and gray are kind of hanging out at the top. No difference between the two, which is good. The red is just essentially like the wild type animals, and we see that we choose, I always forget the dose on here, the 0.3 microgram dose. And we see a similar effect. There's an onset, the animals go back to baseline, and then after they go back to baseline for a couple of weeks, we give them PGE2, and they are sensitized. Interestingly enough, remove the toll-like receptor 4 from the macrophages, you have this refractory effect across the entire acute phase, and the animals don't prime. And this goes back to that pre-transition thought where the immune cells interact with the stimulus to cause the sensitization of the sensory neurons. And it seems as if in this particular context, the immune cells are super important to be activated to cause that sensitization and to cause the hyperalgesic priming in males. However, when you do the same experiment with the females and remove the toll-like receptor 4 from the macrophages, they don't have that same effect, and they actually do prime. Meaning in females, whatever is happening with sensitization, especially hyperalgesic priming, is not due to macrophages causing the sensitization of sensory neurons. Okay, so let's knock out the receptor in sensory neurons. In males, when you do that, it's not as robust, but there is a an inactivation or I guess a slight recovery or prevention of severe pain states and hyperalgesic priming, but it's not nothing. The difference here is between this group and not between the vehicle groups, actually. So there still is hyperalgesic priming to a point, just not as severe. And in the females, though, when you remove toll-like receptor 4 from the sensory neurons, you see pretty much a reversal in that acute pain state and also the hyperalgesic priming state, right? So before, if we were trying to figure out what works in human, what would work in a human, and only do this experiment in males, we would say, well, if we inhibit the immune system, that should do the trick. And then 50% of our patients don't respond. All of a sudden, our clinical trial is going to fail. So it just doesn't work, right? And so this is kind of what we are trying to get at. And so we did this experiment and tried to publish it right away. And people were like, you sure? You made these mice, but do they work? Blah, 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 blah. So we made this re-expression model, and to understand if you have the toll-like receptor 4 only in one cell type, is that sufficient to recapitulate what you see normally? All right, using the same Cree models for the sensory neuron and the immune cells, but instead of having the LOXP sites around the exons, we have this transcriptional blocker that has the LOXP sites, similar to your tomatoes or TD tomato. And then when you have Cree-mediated recombination, you take out the transcriptional blocker and then you turn on that Cree and that particular cell type. And here goes some validation data. One is some gene expression, taking the DRGs that house these sensory neurons and looking at the Cree-negative animals next to wild-type animals and seeing that there is this huge decrease in gene expression. Looking at toll-like receptor 4 and sensory neurons only when you have the Cree present. And then this is a way bigger screen than it needs to be. I need to probably tone this down. Using flow cytometry, taking the dorsal root ganglia, separating out the cells and looking for the protein. And when we do that, here on the top, we see, we can ignore some of this stuff because this is from a different paper. So, but you have your wild type group of normal animals, your reactivated animals, and then your knockout animals. Toll-like receptor 4 and the sensory neurons in your knockout animals is 0. And then when you reactivate in NaV1.8 or wild type, they're right on top of each other with both the males and females. That's what I wanted to show. I probably should just block the rest of this out. But it is important to show it because if I look and want to make sure that the receptor isn't in other cells, we did immune cells down here. And so if you look at both of this, they're in the wild type control animals, there's a 20% upregulation versus nothing in the reactivated animals, but it should be nothing in the immune cells, right? So it works. Long story short, the models work like they're supposed to. You have your re-expression in the cells that you need to, and not in other cells. Okay, so you like that dramatic pause right there? I put down my thing and started breathing a little bit different. COVID hit, and COVID wasn't important because we outsourced this HMGB1 that was redox dependent from a company that was in Switzerland. We needed them to make it. We couldn't make it. And COVID hit. What do you think happened? They stopped making it. And so that really changed the game on us in the context of wanting to keep this story consistent, right? And I usually show this, especially when it's students. They're always like, well, you change your what you're using. Is that the same thing? Well, yes and no, right? We want to use a TLR4 agonist. We want to keep it the same. You can't, right? This $5,000 aliquota stuff that you would buy is now dried up. You can't use it anymore. So what do? You pack it up or do you do something different? Well, we know that toll-like receptor 4 is an agonist, or LPS is an agonist for toll-like receptor 4. And if you get a ultra purified version of it, you know it acts pretty similar to the HMGB1. And we went back to some experiments and validated that it's similar to the HMGB1. And it's a longer story that I don't have time to tell you, but similar enough. And we went forward with that we've gone forward with that for the past 2 years. And so we are now in LPS land. And so that's why I always just say TLR4 induced pain, not HMGB1, because it changes, the story has changed over time. So this experiment is whole body knockouts, the reactivated animal, meaning toll-like receptor 4 only in the sensory neurons, and then a wild type animal. Everybody gets LPS in the paw after baseline, no vehicle controls. I show you because nothing happens, vehicle does nothing. So in the knockout animals, TLR4 does nothing across this particular space. The wild type animals, they look pretty much the same. We chose a dose of LPS that actually, when you look in literature, people have done it quite a bit, so we weren't too worried about it. So we picked a dose in the middle. And then when you see that when the reactivation of toll-like receptor 4 and sensory neurons in the males, this hour here, I'm going to bring up later, but essentially we think it's an acute phase that you need to happen in males, but it goes back to pretty much nothing after a couple hours. Interestingly enough, the females show a recapitulation, meaning the wild type phenotype and the reactivatable phenotype is sufficient, right? They drive each other. And when we look at this, the macrophages, we can see that when you reactivate TLR4 in macrophages, it looks just like the wild type phenotype essentially in the acute phase. And knockouts don't do anything. And interestingly, really interestingly, because for some reason we can't understand for the life of us why there is toll-like receptor 4 on the macrophages in the females, and it doesn't seem to be different. If I go back and show you that validation experiment in just a normal animal, they have similar amounts of toll-like receptor 4, but they just don't respond. It's super interesting, and they don't. And we tried this quite a bit at this point. But the original data set and the new data set fit right on top of each other pretty well. And so we feel pretty good about that. Except when we resubmitted it again, we got some feedback from a reviewer that said, well, you're using this thing, lysozyme M-CRE, which I'm not quite sure if people are familiar with it, but I said it. It's a CRE driver that you find, it's a gene that you find in macrophages, but you also find it in some granulocytes. And I've been saying all along, this is macrophages, this is macrophages, this is macrophages. And people get kind of caught up in that, and you get really excited about something. But the reason why I can say that, or I did say that, is this experiment here, which is definitely my favorite experiment so far. And so what we did here was a double CRE experiment. Had both our NAV and lysozyme M-CRE animals, bred them together with our TB background, meaning whole body knockout background, except for macrophage or TLR4 in the sensory neurons and in the macrophages, right? And granulocytes technically, right? But what we can do is treat our animals with something called clodronate that kills macrophages specifically. So we do our experiment, our experimental timeline, where we go ahead and do our treatments to kill our macrophages and then go and do our experiment like normal behavior. Or we do this other side experiment, which is collect all the different compartments to see if the macrophages are there and other cells are there. Long story short, they are. Looking at my time. On the side here, we look at the paw scan, looking at the clodronate positive group. This is particular CD11B, CD45. There's nothing. And then when you look in the blood, same thing. And the clodronate animals, we're getting rid of specifically macrophages, but the other cells are definitely there. You're going to take my word for it. I don't have time to show you all of it, but I can. Okay, so why did it pop up all at the same time? I don't like when it does that. I like it just showing one thing at a time. All right, anyways, let's ignore the right side for now. So on the left side here, so these are the males, double CREs, but I use clodronate, the black group, to kill the macrophages and give LPS. This group, we decided to kill the macrophages and give vehicle groups just to see if you kill a macrophage, does it cause any kind of sensitivity whatsoever? Because we weren't sure. Turns out, kind of luckily it didn't, and the vehicle group, but then when you just had the animals that didn't have any killed macrophages and the sensory neurons, you see a normal acute phenotype. So killing the macrophages is really what drives this particular phenotype, because toll is on both receptors. Toll is on both cell types, but then we kill one cell type. Here in the females, kill the macrophages, and we still see the same phenotype that we've been seeing this entire time, right? So we're doing things like 4 different ways now. Like, I mean, how many ways can you cook something? And then down here is just some area under the curve things that we thought would be interesting that's not that important. So then what about the machinery that's involved in the transition to chronic pain, the priming stuff now, right? And so if we go back, do the same experiment with our double Cree animals, wait for them to go back to baseline, and then come and then inject them with prostaglandin E2, but kill their macrophages before all this happens, again, you don't see the priming effect versus in the males, but in the females you do. So clearly, we're onto something in the context of this immune cell to sensory neuron communication in the context of therapeutics, but we have a long way to go to understand what's going on and the idea of what's going on in females. So this machinery is something that's now been, I would say, the forefront for about, well, I guess right before COVID. And a collaborator of mine, Greg Dusor and Ted Price, I worked with them for a long time now, came up with the idea that CGRP, or calcitonin gene-related protein, is specifically expressed in sensory neurons and specifically has effects in females over males. So that seemed plausible that that's the driving force, but with the sex types, or with the sex phenotypes. And here we show that toll-like receptor 4 in a different ganglia, albeit very similar, they hit similar cell types. When you activate those particular neurons, it causes the release of CGRP. So it seems, plausible that if you activate toll-like receptor 4, you're activating CGRP, and that's going to be differences between the two. And so we did some experiments where we harvested out the DRGs and peritoneal macrophages from these groups, wild type group, the reactivated group, and the knockout group, the knockout group and the reactivated group. So this is back to a single Cree if you want to know what happens when you activate just the sensory neurons. Take them out, do co-cultures because you want to make sure that both the cells were there. And then we did some ELISAs for inflammatory cytokines and also for CGRP. And here goes a picture of what the co-culture looks like. I just thought it looked really pretty. There you go. And when you look at inflammation and then the CGRP release. So at the top here is IL-6, an inflammatory marker that gets released after LPS is treated, or you treat anything with LPS. There's 2 different groups, vehicle group, LPS group. When you knock out toll-like receptor 4, whole dish, no response. Reactivated, a little bit of a response in the sensory neurons, but then the wild type, everything, you have a pretty good response of IL-6. These are in males. And in the female group, we see this huge recapitulation similar to the behavioral phenotype of IL-6. We were just saying IL-6 does everything earlier. Seems to, right? And this is one of the reasons why we were doing another project looking at the IL-6 receptor, actually. I'm not sure if it's interesting or not, but the CGRP story didn't hold true, right? And in fact, we saw that when you reactivated toll and sensory neurons, there were no differences between the male and female reactivated wild type groups. So clearly there's a different process that's happening. We think it's intracellular signaling and not necessarily these extracellular components, but it clearly has to do with inflammatory mediators and not so much calcitonin-related peptides. So there's a lot more to be done, but I knew I was going to run out of time, so I think I kind of concluded it. Yeah, I did a good thing. And so the conclusions are, or concluding thoughts so far are, nociceptors mediate these sexual dimorphic responses to TLR4 agonists and probably others in general. But macrophages mediate these long-term changes in males, and we need to be thinking about that in the context of long-term clinical application. And nociceptors mediate long-term plastic changes in females. I need to be thinking about that in the other context. but this alludes to a definite difference or dichotomy in the conversation in this neuroimmune crosstalk. And since we started these experiments, there's been 2 papers, one that I'm involved in, one that I wasn't, where someone took out the dorsal ganglia to look at the sex differences between the populations. And what they found, especially in the context of the proteomics, is that there is a divergence in signaling, intracellular signaling, like we were saying. And it looks like prostaglandin signaling is important to drive sex differences. So there's some follow-up to be done there, but that's pretty much where I'm going to end it, because I figured that was a lot of data, a lot of explanation, just a lot of talking, and who wants to talk the entire time? So this is a picture of the lab. Most of these people, not most, so 1, 2, 3 of those people have already, or will be graduating, or have already graduated. Jessica graduated about a year ago, but everybody else is still hanging out with us. For the most part. This is the actual names of the people in the lab, collaborators at UT Dallas, UT Southwestern. We have a couple collaborators on the fibromyalgia project I used to talk about it, but I stopped talking about it. And then some funding sources that we have for the lab, and that is the end. Thank you. All right, questions.
Thank you for that talk. So I have a question. Your opening slide had a beautiful image of the dorsal horn of the spinal cord. And I'm wondering if you had done any imaging or anything looking at, does this translate into like activation of the dorsal spinal cord? Do you see activated or differences in activation of microglia and astrocytes and things like that? Does this move like from the DRG into the spinal cord?
Yeah, that's a great question. I'll repeat it. Essentially, the question is, does some of this activation in the periphery pretty much translate to the spinal cord? The answer, yes. Funny part is I took that out because I knew I was going to run out of time. And so that was actually the beginning of the experiment, where we did it in wild type animals. This neuropathic injury that upregulates HMGB1. And what we saw was in females that they're a little bit more hypersensitive in the neuropathic pain model. This model, not so much. But when you looked in the spinal cord, you could see where those mechanical sensitive or hypersensitive neurons project. There's more HMGB1 deeper in the dorsal horn, and more activation in like microglia and astrocyte activation around the deeper lamina, compared to the more superficial lamina, which had us dig a little bit deeper and then focus more on the periphery with this first part of the story, because that's where the manipulations are with the genetics. But you're actually where we started off looking actually at the spinal cord and worked our way backwards. So it's a great question.
One is a little tangential. So nice talk. So I noticed you had these, get an injection with 24 hours. Saw these changes. I'm just wondering, sleep is thought to, sleep disruption can cause pain-like symptoms. Deep slow wave sleep can actually engage the immune system. So if you looked at how something like sleep might relate to those changes in the dynamics of injection.
So the question is definitely tangential and definitely not something that we normally do, but it's a great question nonetheless, which is how does sleep or disruption of sleep, when you do these long-term experiments, change sensitivity, pain sensitivity? I would say definitely they do. So some of the experiments, you have a 1 hour, 3 hour, 5 hour, 8 hour, and 24 hour. We actually don't disrupt sleep at all. Actually, we do our experiments 1, 3, put them back in the cage. We usually do their experiments in the dark cycle, and then when they're up, right, and then let's put them back in their cage. We actually were just as worried, not just sleep, but fasting. One of my students actually has a paper that's hopefully going to come out soon looking at fasting, which has a huge effect as well. But but we put the animals back in their cages, back in their home cage, so that they don't, that effect isn't as severe. So that it's not like every single time point they're just out, right? It's at the 24-hour time point, they would have been in their cage for 12 hours sleeping or whatever they need to do. So
let me just flip it in a slightly different way. So the idea is that, you can see the humans, if you increase level of sleep, increase immune response and positive. So you wouldn't even necessarily disrupt sleep, right? So one is I guess you inject it, and then there's probably some half-life to whatever you inject it. And you're gonna have some sort of dynamics, right, from 0 to 24 hours. But between that 0 to 24 hours, it could be that the sleep is equally distributed, right? Or there could be periods where there's more sleep. And so the question is it just strictly, those changes that you're seeing? Can that strictly be explained by, the half-life of whatever is being injected or something like that, or could something like, the amount of sleep between those data points also explain some of that data? You don't have to answer it. I guess that's another way of thinking about it. I was just thinking, even if it's not disruptive,
right, yes, I guess I see what you're asking. Like, what exactly kind of explains, or can the differences in the sleep patterns change the dynamics of the behavioral dynamics?
Yeah.
Probably. That's what I'll say. There's a lot of words to answer with a probably, but
Okay.
probably.
I'm just curious because I'm interested in sleep. That's all it is.
Yeah. So we are definitely afraid of changing anything about their cycle because of that. Because if you alter sleeping, you alter cortisol levels. If you do these things at different times of day, you're introducing variability that you want to introduce. So we try to act like we're not changing anything so we can ignore it. So, but we very well be right about a lot of that, a lot of the kinetics. But as long as everybody's treated the same, we feel pretty good about it.
Actually speaking about cycles, so do you have to control for like where the needles are in your cycle?
Good question.
Like, do you think that is their pain or how they respond to?
Yeah, so that's another deathly afraid a question. So the question is about estrous cycle and how does that potentially affect what's going on in the females. So for this study, we didn't do that at all. And I'm kicking ourselves for that because that's like 600 mice per genotype because of what it needs to take to get there, right? And so, yeah. But what I will say though is a lot of studies have now been thinking about this and have shown that if animals are in the same cage, that they usually sync up together, and there isn't a huge difference, especially the differences that the gentleman had the question about the subtle differences between the time points. This estrous cycle doesn't seem to have that effect. If you, especially on neuropathic injury, we do have a follow-up paper in review now. I do have a lot of papers, right? So it's looking at the effect of age, and estrous cycle, and we don't see any effect in more clinical relevant model of plantar and post-surgical pain. So, but you're right, people do it. It's a lot of work to do it. And a lot of people who have been doing it haven't been finding differences, but I wonder why. I wonder if sleep has anything to do with it, because you have to go and interact with the animals a couple times a day, at least to try to figure out what cycle they're on. So a good question. Okay. Have we looked at satellite glial cells at all? Do they express TLR4? So satellite glial cells do express toll-like receptor 4. This experiment, like I said, is already 1,200 animals deep and counting. And so we don't necessarily want to keep going with this, because essentially it's every model or every cell type you want to use, you're there, right? it is something on our mind. I think there's been a couple resurgent papers talking about satellite glial cells, but we haven't really been thinking a ton about them. Maybe we should be, but that's a good question. Oh, okay. And then Jonathan Church says, not sure how off topic this is, but do specific pain sources have unique pain mechanisms that are not observed in other forms of, I have no clue. That's a great question. And actually wouldn't even know how to test that. Great question though. Like how would I ever how would we ever be able to model that? Good question. Even if there are shared mechanisms of pain within the same sex, is there homologous mechanism? That's a good question too. If there are shared mechanisms of pain within same-sex individuals, is there homologous mechanisms of different sexes? So yeah, that's what we're hoping to find out. But really, the first question is really kind of blowing me right now because I don't really answer the question. It's how do I model childbirth in a male and make him feel that? That's what I read when I read that question. I know what he's saying, but you know what I'm saying? Like that's a great question though. That's a phenomenal question in my mind. But
could you put tiny little TENS units on their abdomen to force abdominal contractions?
Maybe.
So I have another very nice question. It's about, there's the incidence rate for these in female to male. A lot of these much higher in female than male. So maybe there's an initial difference, and then the manifestation at the clinical level, I guess, is somewhat similar. But is there also? I'm just wondering in general, does it turn out that actually the clinical differences there are differentiations which across genders as well? It's not just whatever the molecular mechanism is. Maybe they're feeding some common pathway and it's the same clinical manifestation, or is it that actually we've just failed to differentiate some sort of like, for example, fibromyalgia, I know is like huge, like 4 to 1, like 5 to 1, something crazy,
Yeah, I get your question. Essentially, differences are there really sex differences at all? What exactly, because that's a great question, right? The first study was we give everybody HMGB1, they respond the same, right? So even if these mechanisms are potentially different, the ultimate outcome is pain across the board. And so what does that look like clinically? Does that really, that's a great question. And why is it that there's more, air quotes, prevalence, a higher prevalence of pain in females? I actually think that has a lot to do with social factors and psychological factors way more than anything else. I do think, and it does make sense if you think about mechanistically, like neurons driving pain states, that it would make sense, especially chronic pain, that females would experience pain at a higher prevalence. But I think the gap in between the two, in between males and females, that's a whole nother conversation, is way shorter than we think, but still there. And it's because, I used to play football. We were talking about this earlier. Were you in pain? No. Your bone's sticking out your leg. You sure? I'm all right. I'll put it back in, coach. You know what I'm saying? So like, I think there's a little bit of that in there for sure. A lot of bit of that in there for sure. Machismo, etc. So yeah, anyways. If there's more, right? Chat questions. I'm a bad moderator. All right, great. No questions really. Well, they really want to make sure that the satellite question was answered. All right, where's that?
I have one short question.
Okay.
First off, the model you have where you do the priming, if you ever look, or has anyone looked to see how long of a gap you can have to still get priming? That kind of seems to imply that somebody getting an infection could prime them for chronic pain, if I'm reading that right.
It's a lovely question. I love that question. The question, I'll repeat it, is in the priming experiment, we took it out a couple weeks, but really, can you take it out more? Months, years? How long can you take it out? Well, obviously in animals, you got to pay for them, so we ain't doing that, right? But to be honest,
Told you guys have the correct answer.
but so other people, not us, have taken it out, have taken IL-6 out 2-3 months, and you still see a priming effect. It disappears, it dissipates the severity of the priming effect and the longevity of it, but it's still there, right? So that, implicating that it wanes off. Another question that we have, and I'm going to free plug myself a little bit, is do you need to have an actual pain state to have priming? And so, again, I love this experiment, and hopefully it's going to get accepted. Another paper that we have that I'm thinking about it. We put animals on a high-fat diet, but we didn't let them get diabetic. We didn't let them get fat. We just put them on there for a few weeks. And then we gave them PG, we gave them some PGE too. And we found that they do prime after this high-fat diet. But before you get into these pathological states, because the question always is about, especially for diabetic neuropathy, is it the inflammation from the diabetes? Is the dieback of this peripheral sensory neurons from the skin? What exactly is it? And one of the questions that I had was, well, is it just the diet itself that's causing sensitization, period? And it's probably a little bit of all the above. That study is in review with scientific reports right now. So not a huge paper, but like I think is super cool. Another small plug. This paper isn't getting submitted or anything yet, but we have it ongoing, is the effects of alcohol or ethanol too. And this one's going to be sad for a lot of people, especially college age people, is if you put the animals or give animals essentially a beer day or drink a day for about 2-3 weeks, it sensitizes them to a pain state, but it also makes them more depressed when they're in that pain state. So this isn't I'm drinking a ton. This is of having a beer a day, which is pretty normal, right? Or wine, a glass of wine a day, and which is even more normal. And that does cause some sensitivity. And we're trying to figure out some answers. I have a really awesome chemistry student that's trying to figure out some really cool chemical stuff in there. So I'm just going to talk to you guys about that later. But yeah, so the hyperalgesic priming thing is super interesting, and it really opens up the door for really what's going on in this kind of chronic transition. So, okay, so then there's one more question. Ooh, other than sex differences, does genetic background influence pain response? It's not quite related to pain, but I know people respond differently to anesthesia. Yeah, so we were just talking about Jeff Vogel. There's a person in the pain field that's super known for some of the papers that we talked about today, but also some of that kind of work looking at really cool questions like, does the sex of the experimenter change a pain state? Does the breed or inbred or strain of the animal change the pain state? So all this stuff's actually been asked already. And it does. You just have to be mindful of the breed, the strain you use, where it's coming from. And not even thinking about pain, I'm going to use my metabolism background to even tell you all that there's strain differences with C57 substrains in metabolism. And that will definitely change pain states as well, because we know that for a fact. So yeah, this is very, very true and very important. So cool. Good questions. Did I press enter live? All right. I think that is all the questions online. People are sliding out, so I'll let you guys clap for me. Thank you. Appreciate it. Here. Bring your cane. Oh, let me shut this up.
Yeah, it was actually. How to reconcile the different